It's an honor for me, I'm Nora Volkov, Director of the National Institute on Drug Abuse, to introduce today's speaker, which actually has been someone that has made some of the most important contributions in the field of substance use disorder, particularly as it relates to the effects on neurodevelopment. And this person is Dr. Jasmine Hurd, who is Ward Coleman Professor of the Icahn School of Medicine at Mount Sinai in New York. And she's a pioneer and distinguished developmental neuroscience researcher in general, apart from her contributions in substance use disorders. She is a leader in the field of neuropsychopharmacology, brain development, and the genetic and epigenetic mechanisms of substance use disorders research. Some of her pioneer research involved molecular studies of the human brain, including human fetal neurodevelopment. Her multidisciplinary integrated approaches has had a substantial impact to the field in neuroscience and in substance abuse in particular. Dr. Hurd received her training at the Karolinska Institute in Sweden in neuropsychopharmacology. After postdoc research at NIMH, in fact, she took up a junior staff research position back at the Karolinska Institute and was later promoted to full professor in that institute, making history, for she was the youngest female professor at the Karolinska. Dr. Hurd was recruited back to the United States, to Mount Sinai in 2006, and assumed many positions, including the director of the MD PhD program and chief of Center for Excellence in Mood and Motivation at Friedman Brain Institute. She now holds the position of director of Center of Addictive Disorders at Mount Sinai, which serves one of the largest addiction populations in the country. During her career, Jasmine has been highly recognized in the field and published many high-impact papers that are relevant to brain development, genetics, and epigenetics in neuroscience and in substance use disorders. She's also an ardent supporter of students, junior scientists, and minority trainees, and she's an extraordinary mentor. She served as the founding chair of the Diversity in Biomedical Research Committee at Mount Sinai and has been substantially involved in many of the community initiatives. And I would say she is one of the most outspoken about the importance of diversity in the scientific workforce. Today, Jasmine will be discussing her work in two of the most notable current drugs in the United States. Heroin, because of the epidemic of opioid overdose in our country, and cannabis, because of the changes in its legalization status, including its use for medicinal purposes. So it's a pleasure to have you here, Jasmine. But before you come up into the stage, I have been specifically told, all of you told me to tell you, to remind you that after this hair presentation, there will be a reception at 4 o'clock in the library, and all of you are welcome to come, and that will give you an opportunity to interact directly with Dr. Jasmine Hurd, which is really a pleasure. So I really invite you all to take advantage of this opportunity to have this one-to-one interaction. Jasmine, welcome to NIH.
It is overwhelming. Thank you for that introduction. I actually don't recognize myself. I'm wondering who she's speaking about. I loved meeting with the Pratt Fellows today because I started off at NIMH as one. So it feels great to come full circle and be back here talking to you today. So I thank all of you, Dayu Wu, who has been instrumental also in helping bring me along to this day, and Nora, who's amazing for us. So as Nora said, I'm going to talk about two drugs in particular that has been interest in our research, and I was asked to give a provocative title in part. I'm usually terrible at titles, but it was about high times for drugs, and exactly, I think that heroin and cannabis are definitely substances that a lot of our, both in terms of our public audience and our scientific community, are really discussing in large part, and go over about epigenetics and what we've learned in the brain. And for me, when I think about the process of substance use and vulnerability in general, I think the fact that life is very dynamic and our experiences throughout that journey really impacts the behavioral and disorder vulnerability that we have later in life. And drugs really are, for me, this ripple effects of this drops on the water that extends out into different time periods of our life, and including and especially vulnerable periods such as during early development. We know that addiction disorders are complex. They take into consideration aspects of genetics, early development, and the environmental factors that impinge on them. And all of those together alter sensitivities to different aspects of behaviors that we know are important for linking with addiction vulnerabilities, such as these behavioral traits and the psychiatric comorbidities that come to play. For me, we know that marijuana is the drug that is most used in the US, and so is heroin, and the opiates prescription opiates have really come to play in large part in recent years. I'm going to talk to you a lot about the strategies that we use are very translational from what can we learn about the human brain, and we use the brain in terms of our own models to really help provide mechanistic insights. I'm going to start off in large part focusing on the opiates, in particular heroin, which it's incredible the epidemic that we have now in this country. I think a recent number had that nationally about 125 people a day die from overdosing on heroin and painkillers. That's incredible. And it's no longer about the large cities, but has impacted a lot of the suburban and small towns in the US. So we really need to take action in terms of understanding, yes, the neurobiology, coming up with treatments, and using the treatments that are already even there that we're not using enough. And I'm going to start my talk because I'm at NIH, when I was at the NIMH as a Pratt Fellow and later as a staff fellow. This is where my studies in the human brain really began in terms of can we study the neurobiology, the molecular biology of the human brain. And many people told me that was not possible, and I was in the lab of Miles Herkenheim at that time, and he said, go for it, go ahead. And so I thank Miles for giving me that opportunity to say, pick your mentors wisely, but it may not work, but try. And so this is a whole hemisphere, horizontal slice of the human brain. I'm looking at the prodynorphin gene. And prodynorphin is an opioid neuropeptide that's regulated through the kappa opioid receptors that modulate mood, and for the most part, negative mood states. When I was a staff fellow, one of the things that I had found in studying that time when I started studying in my lab in the clinical brain disorders branch of Dun & Weinberger, then developing the molecular components of their imaging big division, was that I saw that the prodynorphin gene is really interesting in where it's expressed in the human brain, and in one part in the amygdala. And the amygdala is a brain region important for emotional regulation. And as I said, dynorphin is an opioid neuropeptide important for negative mood states. And we had seen that in the postmortem brains of people who were diagnosed with major depression or bipolar disorder, but not schizophrenics, that these people had impairment or reduction of the prodynorphin gene in their amygdala. I'd wanted to follow this up for many years and really never did. And MD PhD student Sarah Ann Anderson came to the lab, and we started to, again, study this prodynorphin in the amygdala. And the thing that was intriguing was that we saw a similar pattern of prodynorphin change in different cohorts of people with major depression, but also in people who were substance users. And where we saw this change was in this subnucleus of the amygdala, a really tiny region that no one really studies. It's the periamygdala cortex. And I'm just going to say PAC for short. And whether or not it was heroin abusers and different cohorts, or even cocaine users, I will say as well, this small region reproducibly had changes in the prodynorphin gene. But since we do not know what this region means for the brain, we had to first go to a translational model and look in the animal model and to see whether or not we could replicate these changes in animals. And so we did, and we found that prodynorphin was changed in the homologous region in the rat brain. And it was changed actually at a time period where we saw, for example, the CRF mRNA expression in another part of the amygdala that's important for stress response. So about 24 hours after they had self-administered heroin, we saw a similar change. But what does that mean for the brain? When you're seeing these things in humans, where no one is studying the human brain, and we don't know what it means for circuitry, we decided to try to combine molecular biology with in vivo imaging. And there, I was lucky, Michael Michaelides, who was a postdoc at that time in my lab, he's now setting up his own lab here at NIH, which I'm really happy about. We decided to look at combining DREAD technology, where we would infuse a virus into that area of the amygdala, where we can regulate the activity of these prodynorphin cells and see what it meant to the brain by coupling it with in vivo imaging, and for the most part, FDG microPET. And I think Michael kept on going back and forth, what about calling this strategy this or that? And I think very late one night, about I think 1, 2 o'clock in the morning, he sent me this, let's call it DREAD-assisted metabolic mapping, or DREAM. And I was like, perfect. So what DREAM is, we're able to look at the activity of changing the activity of the cells by infusing the virus, the vector, into this brain region, and we give the animal an inert ligand, CNO, clozapine N oxide, that only activates those cells because it has this mutated receptor that is specific for being active by the clozapine N oxide. And so we look at behavior, we do the imaging, and this is what we found. Just changing the activity of the dynorphin cells in the PAC was very selective to the neural structures that's being changed in the amygdala. We see that the extended amygdala is this network of cells that was activated. This is and into the anterior cingulate. And these regions are really been implicated by Leonard Hamer was a very famous anatomy diet a number of years ago who was amazing for the field, and George Ahid, where they identified this collection of cells termed the extended amygdala that they thought would be important for maladaptive responses to anxiety and stress. And just changing, as I said, only this small group of cells, of prodynorphin cells in the PAC, leads to this very discrete recruitment of this circuit. Not only is it very restricted, interestingly, if you change the activity of these cells, you change physiological levels of cort. So corticosterone levels you can change. You can increase the anhedonic properties of the animals in the sense of they will decrease their sucrose intake, and they show depression-like behavior just by changing the activity of this discrete dynorphin cells. So we know that certain traits, such as negative affective traits mediated by these subsets of dynorphin neurons, are important for substance abuse and psychiatric comorbidity. But when we studied prodynorphin, for the most part, I actually had focused on the striatum, because this region is the caudate nucleus. This is the nucleus accumbens. The nucleus accumbens is important in large part for reward, goal-directed behaviors, and so on, and motivation. And so a lot of our work focused on the striatum. And the work focused also on the striatum because a lot of studies had gone on where we know about in large part, the anatomy of the striatum. So we know that the cells that express dynorphin, they're part of the so-called striatal nigral, depending on if it's from the accumbens, the striatal mesencephalic projections that mediate goal facilitatory behavior. While the other networks of striatal output cells, the medium spinal cells, they will actually express another opioid, proenkephalin. I'm not going to talk really about that today, but also the dopamine D2 gene, which is well characterized in addiction vulnerability. So, as I said, we know that the dynorphin cells are for quote-unquote the go pathway and the D2 for the no-go pathway. And we looked at variants of the prodynorphin gene to see if any of those variants, at least in our heroin abuse populations and a few other populations, where do we find a greater association of variants of the prodynorphin with substance use? And we found it in a large part, actually significant, at least in our populations, in this 3' untranslated region. So because we knew that the prodynorphin was important for go behavior, we wanted to see whether or not polymorphisms of the prodynorphin gene related to your decision making in that sense. So we brought people into the lab, we took them through different tasks of decision making, as I said, learning tasks, probabilistic learning tasks, and looked at reinforcement learning as compared to inhibitory control avoidance learning behavior. And I will take anyone who wants to, in the end, find out more about the tasks that were done. But basically, it's trial and error. You show people certain symbols, and certain symbols they realize are being given, quote unquote, this is good. They're being rewarded with it, not with any drugs or anything like that, but just that this is the good symbol. And after a while of testing, and another symbol, say B, is bad, and they will try to avoid that symbol at the time of testing, and they will choose the A symbol for reward. And one of the things that was clear, and we replicated another group, is that the prodynorphin variant related to your positive reward choices, but not to inhibitory no-go behavior, while the opposite, variants of the D2, related to inhibitory behavior, inhibitory control or choices, and not to reward. So the genetics of those pathways, associated with those pathways, really were in line with the prodynorphin gene. So what does that variant really means on a mechanistic level? The first thing is that there's when you look at, there's a genotype dose effect that is clear for anyone to see, and so people with this G allele show a greater reward sensitivity. They also have higher expression of prodynorphin, and we see greater in the nucleus accumbens, this reward area of the striatum. And as I said, the thing that's interesting about this variant is that it's in the 3' untranslated region of the gene, which earlier many people thought that these were non-functional because they're in the non-coding area. But we now know different. We now know that microRNAs are target this 3' untranslated region to regulate translation of a gene and even sometimes actually in 5' expression of regulation of the microRNAs will regulate gene expression itself. We also know that this particular SNP is only 5 bases away from a predicted target site for a microRNA 365. So this miR 365 is well conserved among vertebrates. So we looked at the miR 365 both in the human brain and using cell culture models as well, and we see that individuals that have the G allele, they actually are more vulnerable to the regulation by this miR. If you have a normal A allele, the miR does not have an impact on regulating the prodynorphin expression. And we replicated that the G allele is vulnerable to being epigenetically regulated. But the causal relationship you have to establish with animal models again, so using animals where they self-administer. Here we just did a palatable for chocolate, and indeed if you change the regulation using here a vector this time that has a synthetic anti-microRNA, you can, which will increase prodynorphin expression, animals will self-administer more reward, palatable food and normal chow, and they'll show motivated behaviors. So we can show and prove that a genetically influenced relationship between prodynorphin and the miR regulation contributes to the positive reward phenotypes that we see in people, and it seems to be mediated by this ventral GO pathway that has been characterized in humans and that we can prove in animal models does exist. I'm telling you about like a candidate genes, for example, prodynorphin. Like I said, I feel like since it started here in part when I was a postdoc, but I'll just take a quick little diversion from all of this to just emphasize that we do genome-wide evaluation of the postmortem human brain as well to really get insights into, in an unbiased manner, what is really aberrant in heroin abusers. And I'm just giving a very short thing. We know that when you look across thousands of genes, you don't have to be a bioinformatician. You can see definitely that there are differences between heroin and control subjects, but the bioinformaticians do help us in being able to analyze all this data. And one thing that is clear is that at least the striatum of heroin abusers has significant impairments of the glutamatergic system. No matter how the analysis is done, they also show significant impairments of epigenetic remodelers. And just for those who are in the audience that don't know about some of the epigenetic mechanisms, they're multiple. I talked to you about microRNAs, but your DNA is either in an open or a closed state based on methylation that will lead to more repressive closed state of the chromatin, or acetylation of certain lysines in the histone tails that will open up the chromatin, and certain chromatin remodeling complexes that are important for open closed state of our DNA. One of the things that we see is that the direction of change of a number of these epigenetic remodelers in the heroin abusers are really reflective of open state of the DNA for enhanced transcription. And interestingly, they correlate very tightly to some of the glutamatergic genes. This is an enzyme that's important for regulation of histone acetylation, and there's not a great correlation in control subjects with, this is a glutamate receptor, but you can see that there's a tight correlation in heroin abusers, and we have identified exactly, at least the region, Gabor Egavari, MD, who's a MD-PhD student in my group, really has been looking at where does this occur. And it's really interesting that this region of the lysine, of histone H3 between lysine 23 and 27 is where we find the significant impairments of heroin abusers. Importantly, the epigenetic changes that we see there are related to the years of use of heroin, so that there's a direct impairment of these epigenetic disturbances with, I guess, each repeated use. We don't have the history of heroin use for everybody, but for those that we have, we see that there's a clear correlation. So yes, there may be a genetic association to start, but there's a repeated use of the drug that changes the epigenetic profile, especially for these genes that we see important for synaptic plasticity. So what other genes and what other systems do we see important for some of the behavioral traits that we know are relevant to addiction vulnerability? So I talked about reward sensitivity. Impulsivity is another trait because the impulsivity is a symptom, or I should say a dimensional symptom that spans a lot of psychiatric disorders, obviously ADHD and addiction. So here we looked at impulsive choice. You can look, impulsivity is a huge umbrella term, and there are many different components of it. When we look at impulsive choice, for example, asking an animal, do you prefer an immediate small reward now versus a larger reward later, as you do with drug abusers, most drug abusers prefer an immediate reward now. Animals are not, they're the same as humans, and as you increase the delay between, if you would get a large reward and you increase the delay to getting that reward, animals that are more impulsive quickly switch to pressing a lever that will give them an immediate reward compared to waiting. And you can look at this discount function. The steeper the discount function, the more impulsive an animal. And as I said, the animal, I didn't say actually, we have them pressing for chocolate. Because just like humans, they love chocolate, so they can either get one lever, they push a lever, they get one chocolate, or push another lever, they get 5. And you start every animal off getting five chocolates. Sorry, they push the lever, get 5 chocolate. And like I said, you increase the delay, and you see how impulsive they are. So we decided to use an animal model that has been well established as an ADHD model for high impulsivity, and they're considered the spontaneous hypertensive rat. They derive from the Wistar-Kyoto line, so we use those animals as the controls. And if you ask these animals, do they prefer a large reward now or later, they're much more impulsive than the WKY controls, and they, like more substance users, would prefer a smaller reward now. They also self-administer more heroin. The red line, they self-administer more heroin compared to the WKY animals. However, this dotted line is you allow animals, one lever gives you the drug, another lever gives you, say, saline. So they quickly learn which lever gives them the drug and will press on that. They rarely, as you see in this blue dotted line, normal animals rarely press on the inactive lever that's not giving them anything. These ADHD models, they are very impulsive and press even on the inactive lever. But they know definitely that which lever gives them the drug, but they're still pressing this inactive lever. I would just want to emphasize that the SHR animals, this ADHD animals, is not just heroin that they like. They also like cocaine. Just so across the dose concentrations of cocaine, they will self-administer more cocaine than the other animals. So we are trying to find out what may be some of the molecular underpinnings for this behavioral phenotype that might give us some insights into addiction vulnerability. So we did a custom array of a number of genes, and one gene in particular came up was CREM. And CREM is a transcription factor, as part of the Cre family of transcription factors. And actually, we weren't even looking, this gene just happened to be on this platform. We weren't really looking for it. But it is one of the most reproducible. Every time these animals are studied, they have reduced CREM in their nucleus accumbens, but it's very specific. It's in a nucleus accumbens core. And a nucleus accumbens core is important for, the nucleus accumbens in general is the most limbic part of the striatum. The shell more for reward, the core more for motor aspects of reward. And when they get heroin, actually the CREM levels normalize. So CREM is reduced, and we then can regulate CREM when we overexpress CREM in the nucleus accumbens core. So, interestingly, it didn't change their impulsive choice, but it normalized, it reduced their impulsive action. Both in terms of in the delay discounting task and when they were self-administering heroin, we could decrease this inactive lever pressing. I'm not going to go into details, but we can also identify that they're epigenetically regulated very differently in the nucleus accumbens core than in the shell, both at the level of histone acetylation. Permissive marks are reduced in this gene, and so is DNA methylation. It's hypermethylated in the SHR region of the CREM in these animals. So the animals gave us insights to saying that we identified this gene. It's important for impulsive action, more so perhaps than just reward. But is there a link to humans? So we went to human subjects. So with Jeff Halperin, a psychologist at Mount Sinai, who has been studying ADHD kids. So these are preschoolers. And we looked at variants of the CREM gene. We chose a variant that was representative of a particular, it's a tag SNP that represented a particular area of the gene. And I'll get into that if anybody's interested later. But we see that in subjects that have this variant, they show greater ADHD, and these are 3 to 5-year-old kids. So already here we see a genetic differences in kids that have been diagnosed with ADHD at this early stage, but they're young. These are teenagers. Again, you see a greater association of the A allele in subjects that are hyperactive and actually measuring just their actual physical movement of their feet. That the physical movement, so hyperactivity, is associated with that. Interestingly, these are the subjects, the A allele, show greater association with drug use. These are about 70 subjects in each group, so we wanted to make sure that in a larger population, so we studied a European population of about 1,054 subjects, we see the same thing. So that the CREM A allele shows greater vulnerability to substance use in these teenagers. One of the things that these longitudinal studies that Jeff Halperin that we've been studying give us is that when they're diagnosed early, the question is, some people actually remit their ADHD diagnosis and others persist. The genetic association that we saw early is actually is being mediated by those people who persist in their ADHD phenotype. So not only preschool, but adolescents, we also had adults who were cannabis users. We see that who show great impulsivity, that the impulsivity is actually mediating, we had seen the genetic link to dependence. So the link between CREM and drug abuse really is about impulsivity. Not necessarily addiction per se. We also see it in opiate users. I won't go so much into that. So for us, we're trying to understand what is it about CREM that maybe give us some molecular insights to, say, target for medications. So we looked at different databases and saw that in some of the databases that quote-unquote represented aspects of vulnerability, that the genes where they overlapped, the one thing that they shared was that they were disproportionately targeted CREM. And this was done in a different strategy than what it used earlier with our animals. And the CREM-related networks are associated with actin cytoskeleton, axonal guidance. So we mechanistically went in cell culture models and saw that when you change when you overexpress CREM in these striatal cells, you completely change the spine morphology of these cells. There's a very profound effect. There's a developmental relationship to it. And perhaps in this, like I said, we came from a very unbiased way of using an animal model, looking into humans, and now back to mechanism. The interesting thing is that this CREM network that we identified, there's a gene in there, LPHN3, which is now called ADGRL3, which we had no clue about, has been highly associated now, more recently, in ADHD studies, in genetic studies of ADHD. One of the most reproducible findings in ADHD populations. So we're looking into this CREM network to see whether or not we can use it to now identify perhaps novel medications that are targeting this network and may be important for ADHD and obviously some aspect of addiction, even though, as I said, the CREM really is related to the impulsivity, not just about addiction per se. So the next part of the talk, I'm going to focus on the other drug that is of interest for us, marijuana. And marijuana in large part just because of the developing brain. I mean, clearly adult use of marijuana are issues that we can discuss, but it's important to understand that a lot of teens and early during fetal development, there are people that are being exposed to marijuana. Marijuana is the drug that most pregnant women use. So what are the impacts of that on the brain? I'm not going to show you today our human fetal work, but just to emphasize that the cannabinoid receptor, these are the cells that express the CB1 where THC and other psychoactive cannabinoids will work, is very highly expressed in limbic brain areas as the amygdala, hippocampus, the nucleus accumbens. This is a mid-gestational fetal development. But our question really is, what are the long-term impact of exposure early to cannabis? And so that's where animal models come in, and we know that early exposure to THC, and you study them as adults, these are animals that will be self-administering heroin. This is an early study where we saw that even at doses of heroin where the animals pretty much take the same amount of heroin as their controls, the animals would run to the lever to get the first hit of heroin much faster. These are adults that never got any more THC than just during the prenatal period. Also, just a small stress will increase their heroin intake. And they take longer, they show enhanced drug-seeking behavior, take longer to extinguish their heroin self-administration behavior. And we also see that they show enhanced motivation. So if you ask an animal to press a lever once for a drug, or here a chocolate, something palatable reward, and you ask them to now press it twice to get that one single reward, and exponentially you keep increasing the work effort. So, and then you see where they will stop working they'll say, okay, I'm giving up. I'm not going to press more than 10 times to get this. These adult animals with prenatal THC, they will work much harder, pressing the lever like 100 times just for one little chocolate, showing greater sensitivity. We have looked at a lot of the molecular changes, and I'm not going to give you the details, but just to say that one of the things that we do see in adulthood with these animals are protracted changes in the morphology and physiological looking of these striatal cells that relate to synaptic plasticity. So our question is, what are the epigenetic mechanisms that maintain not only the behavioral changes, but the molecular changes into adulthood? And so we will look at aspects of DNA methylation. I'm going to come back to that, but first, we were looking at a lot of the enzymes that are important for regulating the histone modifications. And one of the things that came out when we did this screen for all these enzymes that are part of the epigenetic mechanisms of maintaining these processes is MLL1. And MLL1 is a mixed lineage leukemia. It's a histone methyltransferase, also called KMT2A, and it's really important for development. We see that this MLL1 changes are not just during the prenatal period when they were exposed to marijuana, but it lasts lifelong, late into adulthood, and it's also at the level of the protein, so not only at a gene expression. And MLL1 is associated with open chromatin state, and we see this increase in it. We also, therefore, wanted to find out, is MLL1 relevant to even the behavior that we see in the adult animals? And so you can go in and knock down MLL1 in the nucleus accumbens, and you can completely normalize their change in reward and motivation. And this is, like I said, adults over 24 weeks from they had their prenatal exposure to marijuana. We also studied the effects of THC in adolescents, and here we see that they self-administer more heroin as adults. And interestingly, they also show adult animals that have adolescent exposure to THC, they also show this increase in this methyltransferase in the striatum. So even though we talk about there are different properties for this set critical windows of development, at least for THC, we see similar epigenetic changes in the adult brain, whether or not it was a prenatal exposure or adolescent exposure. There clearly will be some unique properties, but we're looking at what are some of the common things, because we do see certain common behaviors. One of the brain regions that we see increased sensitivity to of adolescent THC is the cortex. And we know that the cortex, especially the prefrontal cortex, is one of the latest regions of the brain to develop. And so we have gone in and trying to understand what does THC do to the prefrontal cortical development. And here we use lucifer yellow cell loading to really actually look at the morphology of prefrontal cortical cells in adults right after adolescent THC exposure and in adulthood. So the first thing, again, you can see there actually was no real difference when we looked at, for example, the length of these pyramidal, these are just a cartoon of a pyramidal cell, in terms of whether in the apical tree or the basal tree, no difference in the average length of these cells. But what was changed is the complexity of the branching. So when you look from, this is looking from, in the black line are normal developing animals between adolescence to adulthood, there's no difference in the apical branching here, but you can see that adults with THC exposure, there's a significant difference in the apical branching and also in the basal branching. So there's a shrinkage of the complexity of their prefrontal cortical cells. So a student, Ben Chadwick, who was doing these prefrontal cortical studies, he and Michael Miller, MD, PhD student, I said to them, pick up the pyramidal cells. They're really amazing, and persisted and persevered. And using laser, so we laser captured, picking up individual cells to see what is the molecular phenotype of these cells. So we know that there's something morphologically different. And so when you pick up these cells, then can we sequence these cells? And that's what Ben and Michael have done. And there are some really fascinating things that you see. So these are just looking at I'm not going to give you individual genes per se, but just to tell you what do we see in animals that have exposure to THC. But the first thing is that what do we see developmentally? So from adolescent to young adulthood, we see changes and things that we should see for normal development of the prefrontal cortex. Tangential migration, it's interesting cytoskeletal protein actin projection. But you can see that these are after the THC animals. There's a completely different profile of the gene networks that are in place in adults with adolescent THC. Interesting, if you focus just on the THC animals, you see things related to dendritic development, you see morphogenesis networks that are changed, things that are absolutely, completely in alignment with the morphological changes that we see, that you're seeing this at a molecular level individually in these cells. We also see changes, profound changes within histone modifications, chromatin modification, so that the epigenetic profile is absolutely profoundly disturbed in adults with the adolescent THC exposure. Interestingly, a network that we hadn't expected, but we have hypothesized aspects of stress, but there's definitely the stress network in the adult animals are significantly different. So we do know that adolescent cannabis changes not only the structural morphology of the prefrontal cortex, but the gene network within these individual cells and associated with epigenetic perturbations. And we're diving into this data sets more now. So we know that cannabis is one of those drops that impacts not only the prenatal brain that can last into adulthood and shows this vulnerability, so can adolescent exposure to cannabis. But a question that had plagued at least me, I had a question, I actually didn't think that it would have any effect, was whether or not there may be even a cross-generational impact of this. So we've started studying future generations of animals that had only germline exposure to THC. And the junior scientist in my group, Henrietta Sutzeris, has done an amazing Herculean effort in looking at the offspring of animals using our adolescent model and I didn't give you doses and so on today, but in terms of a low, moderate exposure of THC during adolescence, having them grow up into adulthood without any more exposure to THC, they fall in love, and we have somebody else raise their kids because we don't want to have maternal or behavior impacting their offspring and later on. And so I'll just throw a few things. So the first thing that we saw is that these are adults that were not, their parents were exposed to THC when their parents were adolescents, and we see that these animals will work harder to self-administer heroin. We also see that they showed greater stereotypy to when you're off heroin. There are other behaviors as well. I'm not going to go into all of them, but in terms of they show greater sensitivity to sensory stimuli. We see sensitivity changes in their reward. We see gender differences. I know I didn't talk today a lot about gender differences, but unfortunately, there are gender differences to developmental exposure to THC. But one of the first things that we studied just in a candidate way was just looking at a bunch of genes of interest to start, and we saw that the genes that tended to be changed were those associated with the glutamatergic system, and there are shifts from adolescent exposure from the nucleus accumbens into adulthood. And here we don't see any difference between genders. Both males and females show similar alterations, at least for those genes. We did electrophysiology. The electrophysiological properties of these cells, of adults, animals, as I said, whose parents were exposed to THC, they show electrical physiological properties of enhanced, impaired LTD, I should say. So what are some of the epigenetic modifications that might allow for these things to occur? We decided to look at DNA methylation to start, just because at the time in the field, everyone said to us, DNA methylation is the most stable to go across development, and so we did look at it and did a sequencing for what are some of the DNA methylation changes across the genome. It's a little biased model that we chose, where they're really enriching, looking at more the CPG islands. But one of the things that was clear to start is that a lot of the differentially methylated regions associated with the parental THC exposure was within gene bodies, actually, and less so within the promoter region. And the interesting thing is that where the genes that are changed, we saw a number of the genes have been implicated in synaptic plasticity. For example, and in psychiatric disorders, SHANK1, some of the neuroligin. But when you look at the enriched hubs for where the differentially methylated genes are, there's an enrichment in protein associated with the synaptic, with the postsynaptic density. It's the DLG4, which is postsynaptic density 95. So this is where we see much greater impairment at the epigenetic level in adult animals with parental THC exposure. We're looking now at other epigenetic modifications and actually trying to figure out how does this even happen, not just in the brain. But I want to say it's not only DNA methylation. Just interestingly, these animals also show changes of that MLL1 histone methyltransferase that we saw in adults with prenatal and adolescent exposure. So there may be something to also these histone modifications. So I talked to you as that this is a cross-generational effect or a transgenerational effect, but that was the F1 generation. To really say that this is transgenerational, we need to look in the F2, F3 generations. So this is what you see in F3 generation. Grandkids. So nothing at all could explain, they've never had even germline exposure to THC. Just putting them in an operant chamber that first day and asking them their sensitivity to reward, they self-administer more. And they show greater motivation, enhanced motivation to self-administer these rewarding substances. So germline cannabis exposure does induce these cross-generational effects that we see on a molecular level and also behaviorally. So this is where we're trying to understand, like I said, how this happens. And I don't think this is about cannabis. I think that this is about other experiences that we have in our lives that perhaps that they're profound enough to change some of the epigenetic signatures in germline cells and other things that we don't understand as yet that provides these cross-generational effects. Well, one provides a change during our lifetime to make us more vulnerable in terms of inducing certain behavioral traits like reward sensitivity and impulsivity that makes us more vulnerable to addiction and can even impact the next generation. So with that, I'm going to thank the people in my group who have been amazing. as I said, Michaelides, who now runs his own group at NIH, was really instrumental in developing the imaging and combining molecular biology and imaging, which is, we do need to combine different methodologies. And Sarah Ann Claudia Mars, who's also left the lab now, graduated, and she and Amy did the prenatal studies. Ben Chadwick and Michael Miller are really amazing in laser capturing, individual cells and sequencing to really, for us to really understand what's happening in not just homogeneous tissue, but specific circuits. And Michael Miller also did the CREM work as well. Henrietta Sitzeritz and Corey Watson, the cross-generational effects. And Gavari, a lot of work on the heroin abusers. I didn't talk about any of our clinical studies, but we also run a number of those, and Michelle Yoon in particular has been really great. And I thank NIDA for all their support, because a lot of times we were told that these studies of trying to look at the developmental effects of marijuana, or even studying the postmortem human brain on a molecular level, was not really possible or not interesting enough. And so I thank them for helping me to go down this path and this journey, and I will take questions. Thank you.
Jasmine, thanks very much. And actually, there are 2 microphones left and right. So if you have a question, just introduce yourselves and then ask the question. While someone stands up, I want to ask you a question. And you already prime it and say, I'm not speaking about sexes, but sex was on my brain when you were presenting the ADHD results with the CRAM. And this was obvious because ADHD is so much more prevalence in boys than in girls. So have you looked at gender differences, sex differences?
No, we haven't as yet. But in our animal model, we only looked at the boys, and the human studies were both boys and girls. I don't remember if there were any differences in the boys and girls in the human populations. I have to go back and look. But all our animal studies were done with the boys.
Thanks for a great talk. I was interested in your changes in dynorphin expression, both in terms of the different psychiatric disorders, but also in relationship to the different alleles. And I'm wondering, I don't know if you're aware of the recent Neuron paper that came out showing that depending on which population of dynorphin cells in the accumbens are stimulated, you can either cause a real-time place preference or a real-time place aversion. Work by the Brucas lab. So I'm wondering if you're able to go in and look at the dynorphin expression, the differential changes in dynorphin expression based on these different regions of, say, the shell or the core, to see whether or not, in the human, to see whether or not you see like a decrease in one area but an increase in another area.
Yeah, I thought you were going to say the rat. She's even making it more challenging, the human. But actually...
Or the rat, you can say the rat.
So, he's there, gone. But Michael Miller, MD, PhD student, we're also looking at the pathways of the accumbens cells, those that project to specific areas of the pallidum versus those that project to specific areas of the mesencephalon, and doing the same thing, laser capturing individual cells. And we're trying to look at the profile there. So we'll be able to see whether or not there are certain prodynorphin cells that are, what's the phenotype of that particular prodynorphin cell versus another, and hopefully that'll give us insights. The same thing with the human. The human, we're just looking at one cell in terms of picking up one cell and single cell sequencing, and seeing whether or not we can then start to profile the individual populations of the human striatum. We won't be able to say which ones may be associated with aversion versus positive reward choice, but at least we'll compare it with our animal models and hopefully it'll give us some insights.
Okay. John?
Hi. So, of course, the transgenerational inheritance is really fascinating, and Oliver Rando has a model that of dietary transgenerational inheritance of phenotypes. And he recently published that it looks like there's a truncated RNA in the sperm from the dads that may be involved. And I was just going to ask if you have looked at for small RNAs in the sperm from the dads.
So we have sperm from the dads, and so those are the things that we're, not only for the truncated RNAs, but we have sperm from all the different generations, and that's a major question of how does this happen? We still don't know. So hopefully that will give us some insights.
Wilson?
Thanks very much. Wilson Compton from NIDA. An additional question about the transgenerational transmission of these findings. Some nearly 30 years ago, Ted Cicero couldn't get any support to look at something similar in opiate exposure in the offspring of rats taking opioids. No one believed that it was true at that point. We do believe you.
Thank you.
What do you think the mechanisms are? I'm still struggling because it doesn't sort of gel with my understanding of inheritance.
It doesn't gel with my training either. You know, what we were taught in school, that's why I said to the postdocs earlier, we need to be open in terms of what we think we know. We will see, as John said, you know, is it aspects of truncated RNAs? We really don't know the mechanism by which this occurs. Clearly the things that we thought that, you know, all the epigenetic changes that occurred during our lifetime got wiped away in the germline doesn't happen. I was telling some people earlier that these are the possibilities. Animal models give us an idea of what's possible. I don't know if this completely happens all the time in humans. As I was saying earlier, pregnant women, especially certain socioeconomic communities, we encourage taking prenatal vitamins, so folate. That is important for changing methylation. And so perhaps that's why we may not see something like this in a human. Who knows? We need to study those things. But the animal models definitely tell us that this is a possibility. And I'm glad to know that I'm not the only one who's told, no, it doesn't, it can't work. So 30 years ago, it's sad that it took that long to continue looking into that possibility. But we don't know the mechanism, but there are a number of groups that are trying to look into it. So we'll know soon, hopefully.
Thanks very much.
Jasmine, thanks very much. And I am going to apologize to both of those speakers, but I've been told to be draconian at 4 o'clock in the afternoon, at 4:18. But before we go, I do want to invite you all to come to the reception. It's an opportunity to ask directly the question to Jasmine, and also, very importantly, to get to know her and her thinking process. So I want us all to, again, thank you for a wonderful talk and for being here with us.
Thank you. Thank you.